Welcome back to EQBloom. Today we're zooming inside the cell to visit one of the most underrated organelles in biology, the Golgi apparatus. Textbooks usually describe it in one sentence: the Golgi modifies, sorts, and packages proteins. That's technically true, but it doesn't capture how central the Golgi is to life. If your cell is a bustling city, the Golgi is a global distribution center. It receives packages from the factory, edits their labels, repackages them, and ships them with precise routing instructions. By the end of this video, you'll understand how the Golgi modifies and quality checks proteins, sorts them into specialized vesicles, and supports fast secretion in neurons and immune cells, and how the Golgi failure is linked to diseases from neurodegeneration to cancer. So let's start with the big picture. Here you can see the classic cartoon of the Golgi apparatus, a stack of flattened, curved membranes like a pile of pancakes or pita bread. The Golgi sits near the endoplasmic reticulum, or ER. Proteins and lipids made in the ER are sent to the Golgi in tiny transport vesicles. Once they arrive, the Golgi acts like an international shipping hub. It receives cargo, edits and customizes it, sorts and packages it into new vesicles, and ships it out to the correct destination: the cell membrane, lysosomes, secretory granules, or even back to the ER. Every hormone, every neurotransmitter receptor on your neurons, every digestive enzyme in your gut has passed through this shipping center. If the Golgi stopped working for just a few minutes, your cells would quickly lose control over what gets delivered where. The result would be cellular chaos. The Golgi is not just a random stack of membranes. It's polarity organized, meaning it has a defined entry and exit. On the cis side, closest to the ER, new vesicles arrive carrying freshly made proteins and lipids. This is like the receiving dock of the warehouse. As cargo passes from the cis side to the medial and then to the trans side, each compartment carries out different sets of chemical modifications. We can think of this as moving along an assembly line where each station performs specific jobs. The cis cisternae do early trimming and sorting. The medial cisternae add or modify sugar chains. The trans cisternae perform final processing and start to prepare cargo for export. Finally, on the far side, we reach the trans-Golgi network, or TGN. This is the sorting and dispatch center, the place your later slides focus on. Here the Golgi decides, should this protein go to the cell surface, into a lysosome, or be kept in a secretory vesicle for later release. That directional flow from the ER to cis to medial to trans to the TGN is essential. If proteins move the wrong way, or if compartments lose their identity, the entire trafficking system collapses. One of the most important roles of the Golgi is chemical modification of cargo. This is not just decoration, it's information. As proteins travel through the Golgi, they undergo glycosylation, adding or remodeling sugars, sulfation, adding sulfate groups, phosphorylation, or other modifications that act like barcodes. These modifications change how proteins behave. They can alter stability, solubility, binding partners, and most importantly, where the protein is allowed to go. A classic example is the mannose-6-phosphate tag added to enzymes destined for lysosomes. That little phosphate is basically a GPS coordinate that says, do not send this to the cell surface, route to the lysosome. So the Golgi doesn't just ship packages, it rewrites their labels with biochemical precision. This is why disorders of glycosylation, where the Golgi can't properly modify proteins, cause devastating neurological and developmental problems. The body relies on those sugar codes to interpret where proteins belong. Now we're at the trans-Golgi network, the real heart of the shipping metaphor. In this illustration, you see the trans-Golgi with vesicles butting off in different directions, toward the cell membrane for exocytosis, toward lysosomes, and toward secretory granules that will store cargo until the cell receives a signal to release it. How does a Golgi know which protein goes where? It uses a combination of tags and codes. Number 1, cargo sorted by tags and signals. Proteins carry specific sorting signals, short amino acid sequences, or particular sugar patterns. These act like shipping addresses or barcodes. Receptors in the trans-Golgi can read these tags. Lysosomal enzymes with the mannose-6-phosphate tag are captured by M6P receptors and loaded into vesicles destined for lysosomes. Proteins meant for plasma membrane often have different signals that can cause them to be packaged into constitutive secretory vesicles. Some hormones and neuropeptides are sorted into regulated secretory granules which wait inside the cell until a stimulus triggers their release. Number 2, packaged into vesicles with special coats. Around the budding vesicles, you see the phrase special coats. This refers to proteins like clathrin, COPI, and COPII. COPII-coated vesicles usually move cargo from the ER to the Golgi. COPI-coated vesicles often mediate traffic backward from the Golgi back to the ER or between Golgi cisternae, helping recycle components. Clathrin-coated vesicles are heavily used at the trans-Golgi network and the plasma membrane, especially for sorting to lysosomes and for endocytosis. The coat helps the membrane curve into a vesicle, and it also helps choose which cargo gets included, like packaging specific items into a box with the correct label. Number 3, ensuring proteins arrive at the right place. This sorting process is not optional, it's essential. If lysosomal enzymes accidentally get secreted outside of the cell, or if membrane receptors are sent to lysosomes for destruction, the cell's entire communication system breaks down. So you think of the trans-Golgi network as the FedEx hub of the cell, where millions of decisions are made every minute. This protein goes to the membrane, this one to the lysosomes, this one into a regulated secretory vesicle. That brings us to the next piece. Once vesicles are packaged and labeled, how does a Golgi support rapid secretion? This slide focuses on vesicles fusing with the plasma membrane to release their contents, hormones, neurotransmitters, or enzymes. Here's the basic sequence. Vesicle formation at the Golgi. Cargo is concentrated into budding vesicles at the trans-Golgi network. In neurons or endocrine cells, those become secretory granules filled with high concentrations of neurotransmitters or peptide hormones. 2. Transport along the cytoskeleton. Vesicles travel along microtubules or actin filaments powered by motor proteins like kinesin and dynein to reach the cell surface. 3. Docking and priming. At the membrane, vesicles dock using specific protein complexes. Snare proteins are the key players. They act like molecular zippers that lock the vesicle and plasma membrane together. 4. Fusion and exocytosis. When the cell receives a signal, for example, a nerve impulse or a rising calcium, the vesicle membrane fuses with the plasma membrane, and the contents are released into the extracellular space. So why is the Golgi essential here? It ensures that the right cargo is loaded into secretory vesicles. It regulates the timing. Some cargo is sent out continually, while others are stored for rapid release. In neurons, the correct packaging, neurotransmitter receptors, and ion channels routed via the Golgi allows synapses to function with millisecond precision. In immune cells, rapid secretion of cytokines and toxic granules depend precisely on sorted vesicles emerging from the Golgi. So every time a neuron fires, every time your pancreas secretes insulin, or your immune system releases cytokines, the Golgi apparatus has already done the behind-the-scenes work to make sure that that's possible. When the Golgi apparatus stops functioning properly, the effects ripple across the entire cell. On the left, you see a normal, well-organized Golgi stack. On the right, a fragmented or damaged Golgi, something scientists commonly observe in disease. First, Golgi defects are linked to major conditions including neurodegeneration, cancer, and immune disorders. Neurons, cancer cells, and immune cells all rely heavily on precise protein trafficking. When the Golgi can't sort proteins correctly, these cells lose their ability to communicate and function normally. Second, many viruses take advantage of the Golgi as a secret exit route. Instead of bursting out of the cell, they pass through the Golgi and use its membranes to cloak themselves, helping them escape detection. And finally, when the Golgi misroutes cargo, the cell enters a state of disorganization. Enzymes may be delivered to the wrong place, receptors may not reach the membrane, and essential signals may never arrive where they're needed. So even though the Golgi seems like a simple shipping center, its failure can disrupt entire biological systems, which is why it's so essential for cell health.